My expertise is in breast cancer, in particular how to use the biology of the disease to improve the choice for treatment for patients. So first of all, who is high risk of recurrence versus low risk of recurrence, which you can read in the tumor looking at the aggressiveness of the biology, which I actually developed a test called the 70-gene prognosis signature, also known as MammaPrint, and where we did a huge European trial to show that patients who are clinically high risk but MammaPrint low risk can safely forgo chemotherapy. So that actually with the group from the European Organization of Research and Treatment of Cancer, we did a trial in 7,000 patients providing that evidence, which actually could save up to 40,000 women per year in Europe chemotherapy that they would not benefit from. So that's one aspect of my work. The other one is that if patients are high risk of recurrence, you would want to give them the best treatment. And then my expertise as being a molecular biologist comes in again to look again under the hood of the tumor and see whether the engine in the tumor, besides being aggressive, what are actually the characteristics. So are we dealing with a fast race car, which is of brand type A or brand type B, and then we may have the treatment that if we want to stop that car, that would actually block car type A from driving fast, and another one for driving fast for the brand type B. And so that's actually where we are in the last 25 years that I've been working on this to sort of improve diagnostics of breast cancer in particular, to understand who is at risk for recurrence, and if you are at risk, what is the biology that can help us to decide on the best treatment. So looking under the hood of the tumor, that's a biology you can do in different ways. So every cell and every tumor cell has, of course, active genes. And you can and gene activity you can measure by looking at the messenger RNA. So we've been looking at gene expression, which is a different way of saying messenger RNA. So we've been looking at the gene activity by looking at the gene expression and see which genes are actually active in a tumor where a woman has a high risk of recurrence versus where the tumor and the patient has a low risk of recurrence. And it turned out that 70 genes that were either expressed in the on status or in the off status would determine the high risk of recurrence versus the low risk of recurrence. So what we did is pattern recognition of gene expression by technology called microarrays, and in nowadays more something like, if you heard of that, like RNA sequencing is used. For the 70-gene prognosis signature MammaPrint, we achieved level 1A evidence. So that's now more and more into guidelines, and it's reimbursed by insurance companies. So that's taking its own path, being implemented. And my other work is an understanding who has tumor biology type A or B, so that we can match the most effective treatment to every patient based on her tumor characteristics. So there we are in sort of the evaluation trial phase, but hopefully that will lead soon to diagnostics that can also help to determine what is the best type of therapy. So I think in particular the molecular basis of cancer will drive the treatment choices. And so over the last maybe 15 or 20 years that I've been working on this topic, where it used to be only looking through the microscope and defining a number of characteristics, now we're really looking under the hood of the tumor to look at how's the wiring and what is active and what is not active, and that will determine the treatment. So it's more and more precision medicine, personalized medicine, as we would hope we can achieve, which of course goes in steps. We have not that available for everybody at once, but more and more we understand what patients can already benefit from treatments that are targeted, so to speak, today, and more in the future. So I think since biology and molecular biology in particular starts to play such a central role, one of the important things is actually education. And education of what is all available and how should we value what is actually currently available and how should we design the trials to get new knowledge. So meetings like the IMPAKT meeting are actually crucial to help to a broad audience understand what's going on and get actually that level of education higher.